Hello, my name is Mike Ward. I'm the Global Director of Content at Pharma Insights. We're the publishers of Script and The Pink Sheet. I'm here in Barcelona at the Bio-Europe Spring Meeting. This is where the pharmaceutical industry gets to meet with biotechs, medtech companies, investors, where they talk about sort of the current trends, but also look at options for transactions, etc. It's also an opportunity to get to see new companies that are being created. One of those is Atriva Therapeutics, a company that was created in Tübingen in 2015, and I'm joined by Dr. Rainer Lichtenberger, who is the CEO. So thanks very much for joining us, Rainer. So Atriva Therapeutics, it's what kind of company is it? How would you describe it?
I would say we are, let's say, the next generation of new antiviral therapies for diseases and therapeutic opportunities which had not yet been fully touched by previous and existing therapies.
Right.
And what is new with us, with our therapy, we are targeting targets in cellular host of the cellular host, and we are not attacking viral structures.
Right.
This makes a big difference because the virus cannot escape when it's attacking the body, cannot escape via getting resistance.
Right.
So the body, this is the real new thing with the therapies we have. And this research had been stemming out of two university groups, the University of Tübingen, which is very well known in their microbiology research, the University of Münster, which is equally well known in Germany, headed by Professor Stefan Ludwig, and the University of Gießen. These are the three hotspots of this type of antiviral research. They teamed up, these three professors, some 20 years ago, and did extensively work on the benefits of this new approach. New is at that approach that we are using kinase pathways which the viruses need to upregulate in order to replicate themselves. And we are effectively suppressing that kinase pathways, and so the virus cannot replicate, cannot spread, the disease is suppressed.
So, okay, so you're actually sort of disrupting the ability of the virus to make more viruses.
Yeah, and to spread out, and so the infection is just cut and blocked.
And you say that that's the result of like 20 years, I guess, basic research. Is it an approach that has been tried before, or and you're modifying? I mean,
no, this is an entirely new approach. They worked since, let's say, as I said, almost 20 years on that approach. There is Nature discovery papers behind. This paper was published in 2002. They tried to optimize that approach. At that point in time when I started, there had just been not suitable drugs not available. In the meanwhile, there had been drugs stemming from oncology, the so-called MAK inhibitors, being developed. And we had a chance to took up two out of those candidates from a large pharma company which had been abandoned by them. They created a lot of preclinical data on that in the last 5 years, and we took that up, a team of 8 people among the 3 founders and 5 very experienced serial business entrepreneurs, and created a company around these assets.
So you're repurposing molecules that were actually originally developed of targeting oncology targets.
We are repurposing that for antivirals. We will ask us, oncology drug for just flu? My God. But the good thing is, first of all, it's kinase inhibitors. It's not cytostatic, so much less side effects. The second good thing is we are just using doses 1/10 of the doses you use in oncology. The third good thing is we are just having a treatment cycles of 5 days and not a half a year as in oncology. And the fourth good thing is we have published data that with the doses we are using, there are no side effects observed in more than 100 patients. So we are on very safe grounds. What we have to demonstrate, we did it in animal models, is efficacy. And we can demonstrate that this approach perfectly works, for example, for influenza. We can suppress the outspread of influenza. Still, if you are really ill, if you're really bedridden after 3 or 4 days of infections, this drug will still work. in contrast, for example, to Tamiflu. Right. Tamiflu, you got to go if you have the idea it could be flu, otherwise it doesn't work. Yeah. The other good thing is this drug is working against all known and potential new strains of influenza, because it's really a universal mechanism that all influenza viruses need to follow in order to replicate. The third good thing is we do not have resistance development.
Yeah, I was going to say resistance is a big challenge. So and why is that? Why don't you have resistance?
Because we are not targeting and not attacking the virus and not changing any structures there, but we are switching. We're switching the light off in the cell, if you like. We're just blocking the pathway, we are switching the light off, the virus dies down, and afterwards we're switching the light on again and the cell works.
Okay, so you mentioned that the company's created, there was the academic groups that were involved, you said that there were some serial entrepreneurs who were also involved. Yeah. What about the financial backing? I mean, who's put the money to work at the moment?
First of all, we founded in 2015, and then all the 8 founders put a substantial money behind. Right. None of those got a salary, none of those got any reimbursement of cost. We just was our baby, and we brought it to initial preclinical development step. Yeah. And then in late 2016, we closed the Series seed round of a total of 3 million. 1.5 of that is already signed and paid in, and the rest is currently being closed until April. So the entire seed round is 3 million. Having said that, this is enough. This is the budget to drive our lead candidate into first in man.
Right. Okay. So you're at this meeting. Is the plan to meet investors, or are you looking for partners? Because it's quite early, isn't it?
It is early, but there is a lot of interest already around. We have investors. The current investor base are private wealth and are entrepreneurs, and we are backed by the German high-tech Gründerfonds, Europe's largest seed fund. But we have raised a lot of interest already with larger venture funds. We are currently starting to keep them in the loop in order to close a Series A round. The Series A should have between 5 and 12 million, depending on how far we get, to close the Series A round by the end of this year. This gives us enough fund in order to bring the development to a phase 2 proof of concept.
Right. Okay.
This is what the achievement, and this is planned for early 2020. So in 3 years from now, we will have a proof of concept.
And so partnerships, are you holding that off until you have that proof of concept, or?
No, we have already some pharma companies interested in our technologies, which we keep informed and are following. And in addition, we are using that format, of course, to approach providers of CROs for preclinical, for pharmaceutical, for clinical. This is the majorities of calls I'm having here, which is quite efficient for me. So I have more than 40 meetings alone. Next time I bring my scientific people as well, but we are such a small team right now, only 5 people, that everybody has to do his own job.
Sure. And the IP, because you mentioned that the molecules are repurposed. So where does the real IP of your business reside?
The IP resides with the company. The scientific founders filed early enough with good scientific data a series of 4 patent families protecting use and application of MEK inhibitors and other kinase inhibitors like RAF and MF-kappa-B against a broad series of antiviral diseases. And these are 4 patent families, and the last one will only expiring in 2035. We have just got a US patent approved, which gives us protection in the United States until late 2033. So it's quite a solid patent base we are working with. We are adding this now with formulation patents. We are adding substance patents to that for the pharmaceutical salts, for the pharmaceutical morphs. So at the end we will have a solid patent portfolio from substance up to application.
And presumably that's going to give you a platform, therefore, to generate more ideas and different viral diseases.
This is actually exactly what we are planning. Our lead project is influenza, but about 25% of the funds we got are reserved to very interesting pipeline projects. First of all, RSV. We know that RSV virus is strongly suppressed by that. The next one is Hunter virus. Hunter is a real killer in Latin America, United States, and it is an orphan drug, and there's an interest from US BARDA in that approach. And another interesting target is MERS, the camel driver's disease.
Sure. Yeah, sure.
And these are the projects we are following, actually. Some of them are supported by NIH funds, some are supported by other funds of European agencies. So there is some interest around.
And will you need to, I mean, because your first generation of products are repurposed medicinal chemicals already made. Are you going to have to ramp up your own medicinal chemistry to make new molecules?
in setting up our strategy for that. And we have also had contacts here already with some providers, and there are quite a few experienced around. So in 2 years from now, we will have an own pipeline.
Excellent. Well, Rainer, thanks very much for coming and sharing the story with us.
Thank you very much.
Cheers. Thank you. Bye-bye.